Welcome to today's video lecture on protein synthesis. Today we're going to talk about the steps in protein synthesis, which starts with transcription. So please refer back to the RNA video if you need to refer back to what transcription is and what it does. Translation being the second part of protein synthesis, which we're going to talk about today. What's the difference between codons and anticodons? How do you read a codon chart to turn something into an amino acid chain? And finally, what is a gene expression? What does it do? So, again, the first step in protein synthesis is transcription. This is where we take our DNA and we turn it into mRNA. mRNA are what are called codons, and we're going to refer back to that in a little bit, okay? But the term codons, when we talk about codons, we're referring to our mRNA strands. Then we're going to talk translation. Translation is taking those mRNA molecules and actually turning them into an amino acid chain. Okay? So, over here, we have an mRNA strand, being our long strand of RNA down here at the bottom. Then we have our ribosome here, okay, in blue, which is around two different codons. So two sets of 3 bases of mRNA. Each set of 3 bases of mRNA is called a codon. Okay? What happens is this ribosome takes and pulls in a tRNA that matches the mRNA strand, or is complementary to the mRNA strand. So we have A pairs with U pairs with A, and G pairs with C. So this tRNA strand codes for a specific amino acid. The second codon codes for a second amino acid. So we have CAC, which pairs with GUG on the tRNA, and it brings in a different amino acid. Those two amino acids get linked together, okay, by a covalent bond, and then the ribosome moves down the strand. When it moves down the strand, it's going to release this tRNA so it can go pick up the next amino acid that it needs, okay? And this amino acid strand is going to get longer. It's going to read the next codon, it's going to bring in a new amino acid, they're going to get attached, and then it's going to move down the strand one more codon set. The tRNA pieces are what are called anticodons, so they're opposite of the codons, okay? So we have our ribosome, we have our mRNA strand, which are codons, we have our tRNAs, which come in and bring in the anticodons and attach to the amino acids. The next question becomes, well, how do we know which amino acids they are? To know which amino acids they are, we use a codon chart. It's an mRNA codon to amino acid chart. And the first thing you need to do is know how to read this chart. To be able to read this chart, we actually read it from left, top, right. Okay? So the first base that's in whatever code that we're looking for, we're going to read from this side. Okay? So let's say that our code that we're looking for, the first code that we're going to look for, let's say that first code is going to be GAC. Our codon is GAC. So what we're going to do is we're going to look over here for our Gs. Our G is right here, so we know our amino acid must be in this bottom row somewhere. Okay? So our second base, we're going to read from the top, UCAG. So our second base is an A, so we know it's going to be in this column. So our row and our column intersect in this box right here. So our amino acid has to be one of these 4 amino acids down here. So then we're going to read the last one, C. C is over here, where each of these boxes has UCAG in them. This is our third base row side. And what you're going to do is you're going to figure out the row that you're in, and you're going to read down UCAG So we're looking at C, so it's going to be the second word down. So then we go back to the box that we know it's in. It's the second one down, and it codes for the amino acid aspartic acid. So being able to read this chart is the first step. The second step is knowing that there's something called a start codon, and there's also something called stop codons. So within our amino acid, our mRNA chain, every protein is going to start with the amino acid methionine. Methionine is coded for by the mRNA strand AUG. AUG is the start of every single mRNA strand that we're going to code for. So, whenever you look at a long strand, you have to read all the way across until you can find an AUG, and then that's your start. That would code for methionine, and then you'd read your next set of 3 letters. Figure out what they are, read in the chart, and go from there. So then we go from our this is our mRNA codon to amino acid chart. Once we have our chain of amino acids, actually let's go back real quick and take a look, because there are 3 stop codons also. So how do we know that it's the end of something? In a sentence we write a period or an exclamation point or something like that. In the mRNA code, they have what are called stop codons. You'll notice that there are 2 here. The UAA and UAG both code for stop, along with this one right here, which is UGA. Those all code for stop. So what's going to happen is when it reaches that, there's not going to be an amino acid attached to the tRNA, and the ribosome is going to know that it's time to release the amino acid chain. It's going to release that amino acid chain. Once it does that, then it's time for the genes to become expressed. And so going through gene expression, first of all, we have our proteins made. It's the DNA turned into mRNA, turned into amino acid chain, which happens for the most part in the ribosomes. The ribosomes are the structure that makes the protein. If you remember back to our section on cells, you'll remember that the amino acids get folded correctly to allow for the best expression of the proteins inside the endoplasmic reticulum. Okay, so now we're going to take that long chain of amino acids, and it needs to get folded into a correct shape. Once it does that, it gets transported from the endoplasmic reticulum into the Golgi bodies or Golgi apparatus. And that's where additions are made to the proteins if it needs to happen. Maybe a carbohydrate chain needs to be added to the outside of that protein, and it's going to become a receptor protein for the outside of a cell, of the cell membrane. Okay, so any additional pieces to that protein are going to be added here, and then they get packaged into a vesicle so that they can then get sent outside wherever in the body they're needed, okay, to show you who you are. So for instance, you have specific proteins that produce pigments, or that are pigments, that help color your hair, the specific color that it is, or help color your eyes, the specific color they are. Different proteins do different things within your body, help your body function the way that it needs to, and so then you can be who you are.